printing with cells, like we mentioned before, all the techniques that we presented on this morning in our introduction to bioprinting are compatible with cells, although that's a very general statement, and there's of course many times when they're not compatible with cells. So we'll focus on those methodologies which we can include cells and how we might support cells better during our printing process. And so in considering the inclusion of cells into printing, we mostly think about directly adding cells into a hydrogel. So in this case, we have a gelatin that we're just printing out, and there's just individual cells that are distributed within this a hydrogel. That's definitely the easiest, simplest way to include cells, but not necessarily the most conducive to having a structure that is biofunctional and with high cell viability. You might also consider, instead of isolating cells individually, it's taking cellular spheroids and using cellular spheroids, suspend that within your hydrogel, or perhaps stronger is take extracellular matrix in a mature tissue and use spheroids of cells in extracellular matrix. In a similar vein, several different groups have used cellular pellets, taking a chunk of cells that have been before resuspending them, and instead of resuspending them in a hydrogel, using the actual individual pellets of cells and printing with those. Other groups have used an extrusion printing methodology in which they actually, instead of printing cells in any material, they just take a tissue strand that they've dissected and use that entire tissue strand as your printing fiber in extrusion rather than using a polymer print head. And of course, we can use decellularized extracellular matrix and incorporate that with cells as well to help provide some substrate for our cells during the printing process. When we're designing our Bioinks, I said before, there's a lot of compromise between the printability and the biocompatibility of the Bioinks, and traditionally that's been something that we've struggled with for several years. And we call this, here our biofabrication window, where we're trying to find materials that produce the most functional cell populations possible with increased cell adhesion, while also ensuring that we have a structurally sound scaffold that meets the resolution requirements for whatever tissue and whatever CAD model we're investigating. These traditional Bioinks incorporate a lot of the characteristics that we mentioned before, ranging from viscosity, which affects not only the uniform cell encapsulation, but then also the shear thinning effects on cells, which might affect cell viability. The gelation kinetics that we talked about affect structural fidelity, as well as can affect the cell's distribution within our gels. And so all those factors that we talked about earlier today are going to come into play here too in our traditional Bioink formulations. But it's worth it also to consider some more advanced strategies to move beyond this kind of poor compromise between resolution and cell viability and function and start getting into more readily active materials. So for instance, we might choose to use a multi-material Bioink in which multiple materials directly interact with each other. And in this case, we're using two different materials that are printed and cross-linked at various junctions that might be different functional groups that are directly incorporated into these materials. Or we can use an interpenetrating network in which the different polymers we use actually don't interact directly, such as if we use gelatin and alginate, but they cross-link within their individual polymers in such a way that they entangle with each other through these networks. We might also use nanoparticles. There's a wide range of application for nanoparticles in these materials, whether we're using them simply to adjust the viscosity or the mechanical properties of our materials, or we're including more bioactive particles along the lines of including growth factors and drugs eluted or attached to these nanoparticles. And then finally, going back to the cross-linking methodologies, if we have polymers that have non-covalent binding domains, this gives us a very dynamic polymer. So if you think back to those non-covalent bonds that we talked about this morning. All of that can drastically affect the mechanical properties of your bioinks under different stress conditions. So we can think about these different techniques to start moving beyond simply a permanent traditional scaffold and have much more control over some of those effects on biocompatibility and mechanical strength and resolution of our prints. It's worth considering all the different ways in which printing can affect cell properties and cell viability. So for instance, dispensing pressure increases the shear stress with which the cells experience. We obviously know that that drastically affects the viability of cells due to the wall shear stress on the wall of both the nozzles and the cartridges these are being printed out of. Nozzle diameter decreases might affect the cell viability as well, because that also affects the high shear stress experienced by our cells. And then it's worth it also to consider the chamber temperature, either the temperature which you're keeping these inks, these bioinks during the printing process, or even the temperatures at which these cells are, or the bioinks are curing, because, of course, cells are extremely sensitive to temperature. And so understanding back to some of those first points where we're talking about shear stress and needle geometry, we need to know what's going on here at a fluid level. We saw earlier a little bit about this, but we are, again, experimenting with non-Newtonian fluids for the most part. So you can expect to see kind of this laminar flow profile through the nozzle tip. And if we consider what the cells are experiencing at this point, we have cells that are translating vertically down these center lines, but they're basically they're tumbling around here. They're experiencing different shear stresses depending on their distribution throughout the needle. And the higher concentration of cells and the narrower the diameter, the more shear stress is going to be experienced by these cells along the nozzle wall, which will drastically decrease cell viability. If we compare the concentration of cells, then, for instance, with the pressure that we're dispensing cells, you can see that with higher pressures, as we would expect, we greatly decrease the viability of cells. So even though we're increasing these cell concentrations to pretty significant amounts, the largest increase in reduction, or the largest reduction of cell viability, is seen by altering the dispensing pressure within our bioinks. We also can see that the concentration of the bioink, which I haven't talked about too much, will impact the viability of cells pretty significantly. So this is one of those challenges where when you're trying to print with the bioink for the first time, you'll often tend to go towards a higher concentration because you have higher viscosity, you have better cross-linking potential, you have better structural integrity, you have a product that usually, like I said, gels faster, so you don't have this spreading effect of your bioink. But by doing that, you sacrifice the viability of your cells extremely quickly. And in fact, the biggest determinants of cell viability in extrusion printing methodologies often comes down to the concentration of cells that you're using in your bioink, as well as the dispensing pressure that you're using on your bioink. So if we want to protect cells, besides looking into adjusting those printing process parameters, there are things we can do to the biomaterial that we're using as well that helps improve viability. In this case, one of the most obvious examples of improving cellular viability and attachment is RGD modification. RGD, of course, is a very commonly used protein in tissue engineering that's found in the extracellular matrix that allows cell binding. So simply by incorporating RGD into alginate, which alginate, as I mentioned, is a plant-based hydrogel, and plant-based hydrogels generally do not support cellular attachment. we can all of a sudden see a significant increase in cell spreading and adhesion, which will encourage the long-term viability of these cells over time. So in this image, normally, instead of these nice, long spread green cells that you see here throughout the alginate structure, you would see balled up cells that within the alginate. So just by incorporating something, a strategy as simple as RGD or other adhesive proteins, we can improve the long-term outcomes of our Bioink-based scaffold. Now, one other method that we might consider, as I mentioned, increasing that alginate concentration. You saw how just moving up to 6% alginate in solution can impact the viability of our cells. We may be interested in blending different polymers together in order to get the effects that the viscosity effects we might have with 6% alginate, for example, without reducing the viability. So you might use a gelatin-based Bioink, for example, and add some alginate to that, and in effect, you're blending these two different polymers, which provide not only better viscosity for printing, but you also actually add functional groups for cell adhesion and function through the gelatin. So I mentioned that before. It's really important to consider whether or not a single material is going to be good enough for the job, and if not, then start looking into using blending different biopolymers together. Yeah.
From this and what you mentioned earlier, if you have a two-part polymer system, a long and a short crossing, with the flow through one of those syringes, do you run into trouble with one advancing faster and one getting caught in the sidewalls, or is the bigger problem the shear that you mentioned, or are neither really affecting?
Yeah, so if we're using two different materials, is it a problem on some of the printing effects like our shear thinning, or whether or not we're actually getting the materials advancing at the same rate? And yeah, that is actually a big problem when you're using two different materials. So if you're not directly blending the materials in one syringe, you do sacrifice some of your printability depending on the printer hardware. So this is all going to be dependent on whether or not, for instance, if you're using a multi-material head, whether each of the different material chambers has different controls in it or not. So that goes back to a hardware consideration. And you may be limited if you don't have control or individual controls over these different compartments within the same head, then absolutely you'll be focused on trying to get the viscosity of your alginate component to match the viscosity of your gelatin component. And if you don't get those concentrations and viscosities to match up, then you're going to have pretty significant problems with printability. On the other hand, we've successfully and honestly pretty easily incorporated both gelatin and alginate into the same printer head, and then your printability actually increases there from going with just gelatin or alginate alone, because you have a couple different cross-linking methodologies you can use, and you also don't have to incorporate a high concentration of either polymer. So finally, moving right along with the incorporation of RGD and other functional proteins, we can go ahead and include the entire decellularized extracellular matrix as a printing material. The one challenge with using extracellular matrix as a cell-containing printable material is we often see that it has very low viscosity and then as a result poor resolution. So even if you have these really nice fiber strands of polycaprolactam or another gelatin, you have to accommodate for the fact that extracellular matrix, whenever you're incorporating that into or using that as a Bioink on its own, will have kind of some of those pooling effects that we mentioned with a low viscosity material. But that can be a really viable way of designing a structure so that you can catch that pool of extracellular matrix with cells, and then you avoid a lot of these challenges with the materials themselves by using the extracellular matrix in lieu of trying to incorporate a bunch of different growth factors and making all of that printable within your construct. Of course, I mean, it goes without saying that the temperature that's necessary to print your BioInk is going to drastically affect the viability of your cells. And unfortunately, a lot of the materials that you're going to use in bioprinting, you're not going to just use them at 37 degrees Celsius. So you have to find a compromise between the temperature that makes the materials printable, as well as not just in the printing head, but also in this case, this x-axis here is the chamber temperature, or the temperature of the base plate on which you are printing your materials. So while a material might be liquid at 37°C, when it hits the substrate, you want that material to gel, so you might have cooled down the printing substrate. If you've cooled down this printing substrate to something like 5 degrees or 20 degrees Celsius, then you're going to, of course, affect the viability of cells there. With that being said, we can still print something where cells are printed in a material that's only at 20°C and is printed on a substrate that is at 5 degrees C to encourage gelation. And if we give that construct time, we do get full recovery of the cells because in this case, at least, the gelatin that we're printing with has some kind of side or it protects the cells a little bit from that thermal stress. So again, that goes back to always considering, while we're making these compromises with things like temperatures and the printability of our BioInk, what we're really considering is, how that scaffold will perform over time and what kind of post-processing techniques are necessary to make our BioInk functional. I don't want to get too deeply into all the different methodologies that we can use to incorporate vascularization, but it goes without saying that you absolutely need a strategy to encourage vascularization when you're 3D printing any of these substrates. So that's another thing that often gets forgotten when you're designing a scaffold, is you might have the perfect biomaterial and the perfect setup to encourage something like bone growth or tendon growth. But if you don't have any type of vascularization or any kind of diffusion within your cell organoid or your biofabricated scaffold, then all that work was for nothing. So whether you're just incorporating growth factors into your scaffold that will encourage the ingrowth of vessels, or you're directly designing through your 3D printed or through your CAD model different vessels that you can add to your scaffold, you have to have some strategy there. So in this case, there's a variety of research out there that you can look into, whether that's, like I said, directly incorporating the cellular components of microvasculature within your scaffold, or building these sacrificial scaffolds that can be washed away and perfused with the relevant cell types after you've produced your scaffold. So there are several really unique and interesting strategies out there to encourage vascularization, and I think the easiest way to identify which one of those will work for you is, of course, identifying which printing process you're using. And finally, the very last consideration, which we've kind of mentioned before in printing with cells, is that you have to consider the environment in which this printer is housed and where you're actually doing the printing itself. So, of course, sterility is necessary, and there are some printers that will work really nicely within a hood, and there are other printers that aren't going to play nice with the hood. So you've got to ensure that you've got some method of at least isolating and reducing contamination in your cells. Time outside of culture is going to depend on the cross-linking time, the exposure time, all those factors that affect printing time, of course. And as I mentioned, in the print heads themselves often function beyond or below physiological temperatures, as does the printing substrate. So all that will affect the temperature of your scaffold and of course, affect the thermosensitivity of your cell populations. And another thing that is often neglected in a lot of research papers and a lot of talks on bioprinting is how are you keeping the cells within your printed constructs hydrated, and how are you supplying nutrients before and during the printing process. So if you think about it, you've got cells sitting in your print heads for a while. You've got cells sitting on the base plate for a while after printing. And again, like these seem like common sense things, but all of these things are worth considering if you want to produce the most biofunctional scaffold possible. So that concludes everything on printing with cells that we're going to talk about for now. And are there any other questions on that subject? So I'm going to briefly introduce direct electrospinning or direct right electrospinning on this last presentation. This is something that we are going to have the capability to do in the biomaterials lab, and it's a really interesting biofabrication technique, though generally you aren't going to include cells on it. I think it offers a lot of potential. Traditionally, there's a lot of research that's already been done with electrospinning, and I don't need to reiterate all that here, but the major benefits are that you can have fibers on the scale of nanometers to micrometers, which is fantastic when you're talking about cells, because that's a very friendly scale to design our architectures on if we're supporting cell attachment, infiltration, and migration through a tissue engineered scaffold. In electrospinning, traditionally relies on using a polymer solution that is drawn out of a syringe or a reservoir up here, and then there's a potential difference or voltage between the spinneret and a collecting plate, which overcomes the surface tension of the material that's coming out of our spinneret. Now, since traditional electrospinning techniques rely on solvents to accommodate the polymers, we get this really random whipping formation as that solvent starts to evaporate in the electrical field generated by this potential difference. And because of that, you get these random non-woven meshes. There are some ways that you can control the direction of the meshes produced through electrospinning, but none of this really competes with the precision and accuracy that we can get through direct write or melt electrospinning writing. In this case, we're not using a polymer solution, but rather a polymer melt. So you'll see a lot of polycaprolactone applications, for instance, because all those same materials that we can use in extrusion polymer printing, we can use in direct write electrospinning. So this makes it a really natural transition from a lot of the materials we're used to using in bioprinting into and using them here. In this case, because we don't have any solvent evaporation, we can actually produce a really uniform jet of material, and we can have these very nice deliberate designs that are very similar to the scaffolds that we're going to produce with our extrusion bioprinting methods. We don't have any charge density to work with the since we don't have any solvent that's evaporating, which gives us which reduces also the bending repulsion, which we'll talk about in a little bit here at the interface between our collector and our material. So we have this really, really nice fine jet that comes down onto our collector, and that's exhibited here in these images. So here we have two different methods of the direct write electrospinning compared to what we might usually see with the traditional electrospinning that relies on a solvent or a polymer solution. So again, what we're focused on doing here is capturing some of those electrospinning techniques to fabricate patterned nanofiber mats. And we're not talking about just deciding the direction of these fiber mats, but we're able to move in the full three-dimensional space here to design scaffolds that are on the nanometer level. And this is this technique drastically improves excuse me, some of the scaffolds that we see in electrospinning, but it also comes at a cost. So while we're capable of producing those same electrospun fibers at these cellularly relevant levels, it's very easy to introduce defects into the fibers. So you have to use a lot more fine tuning on the voltage difference, the speeds at which the materials are ejected from the spinneret, things like that. And we'll talk about that in a second. But that being said, if you can control those parameters, then here's just a few more examples of the fine microarchitecture you can achieve with direct write electrospinning. So I just want to very quickly show one example, or maybe not show an example. Well, in any case, this is a commercially available 3D printer that traditionally uses extrusion printing methodologies. So in this case, they've 3D printed a gelatin substrate here, and then through electrospinning on the same printer, you can create a directed woven mesh in the center here. So we have not only the mechanical properties on a macro scale of this larger construct, but you can directly electrospin your fibers from polycaprolactone or gelatin or whatever you're using as your polymer melt into this scaffold. So you can combine this printing methodology with other methodologies. And that's something that we'll be investigating more in the Biomaterials lab if that kind of nanoarchitecture is at all of interest to you. And like I said, the advantages here are we have much more relevant microarchitectures than we can possibly achieve with traditional extrusion methodologies. But of course, we can't include cellular components or other bioactive components because of the relatively small polymer jet that we rely on to produce uniform fibers and the temperatures required to achieve our polymer melts. So some important considerations here when we're producing these nanofibers. Like I said, they're really sensitive to tuning the right properties. That includes the speed at which the spinneret moves across the collectors, as well as the velocity of the collector itself. This is if the collector isn't moving anywhere. If you've got a stationary collector, you get this heel that you produce with the fibers. This is something that you see on extrusion printers as well, but generally this error isn't as obvious as it is in electrospinning since you work with much finer fibers. So what we call the critical velocity or the critical translation speed here is when the speed of our collector is slightly increased compared to the speed of the jet itself. And if we can do that, we can start to achieve more uniform fiber deposits, which will produce more predictable fibers on our scaffold. If you increase this speed too much, then you can see how you start increasing that length of that heel, which is going to make the ends of each layer wildly unpredictable. So if you don't get these type of factors and process parameters really fine-tuned, then you can end up with errors that start propagating extremely quickly. One of the main goals, besides tuning the collector speed and the jet speed, is that you want to have uniform mass flow rate through the spinneret. So whereas you might use a syringe pump for the traditional electrospinning methods because you're just getting this random fiber anyway, you may not be able to use a syringe pump for direct write electrospinning because you need a little bit more accuracy and uniformity in your mass flow rate through the needles. Otherwise, you get these pulsed fibers of uneven diameters. Some direct write electrospinning techniques use kind of a screw head, so that way you're providing a uniform pressure that's constant throughout the extrusion of these fibers within your construct. You can also see, because we're using such small fibers, these are problems that you might see in extrusion printing where you get these dips in your construct. But if you don't accommodate for those very early on, then these dips propagate wildly within even just a couple microns. And so there's no way that you can take direct right electrospinning and produce a very large construct if you can't, reduce these errors on a much smaller level. So like I said, besides worrying about things like the collector speed and the speed of the jet coming out of the spinneret, the next most important aspect is incorporating uniform mass flow rate through your ejectors. So that's my very brief introduction into direct right electrospinning. And the major takeaways is that while this is a very sensitive technique towards processing parameters, there's very high resolution capabilities here that you can't achieve with any other type of biofabrication process. And we're able to incorporate this because of the unique attributes of these polymer melts. And we can incorporate this into other scaffolds and other bioprinting techniques. So I'd encourage you to explore it if you want to move beyond a lot of what's been done with the traditional extrusion techniques and see if it's possible to incorporate these direct right electrospinning techniques into other constructs, or even use them on their own in scaffolds if you can get those parameters tuned properly. So that concludes our talk, our very brief talk on direct right electrospinning. Any questions on that? Yeah.
Did you have any electrospin systems here at Rice?
We do. We have normal electrospinning that's available, as well as we will have direct right electrospinning coming up here in May. The company that makes the traditional electrospinning methodology is Y-Flow, and there's nothing fancy there. You have a choice of rotating mandrels or flat plate collectors, as you would expect with, like a homemade system. And then the video that I was hoping to show is a Swiss company called Regenhue, and they provide extrusion-based printers, and they've just come out with the direct right electrospinning head. So that's where we can now easily incorporate our traditional bioscaffolds with this direct right electrospinning technique. All right. So thank you very much for your attention, and thank you for sticking around for these different lectures on the basics. In the afternoon, like as soon as you guys come back from lunch here, please plan to be here around 1 again. And I promise that's going to be more fun, getting to see the actual projects and getting to see people printing stuff and working with some of the tools upstairs. So if you didn't bring your own lunch and you're not familiar, we do have a cafe that's right around here on the second floor. Otherwise, there's a couple places across the street that you can grab food. Anything else that I can address before everybody goes and gets food? Yeah. You know what? I'm sure there are. I'm not super familiar with that area, but I'm sure you're right. When we're working with nanoscale fibers, we introduce a host of other nanoscale problems with those fibers, especially as they degrade, both systemically and locally. But I don't personally have a good answer for that. But again, I have some of the resources on those slides have much more thorough explanations of both the short-term and long-term effects of these nanofibers on cells than I have.